scientists were editing DNA decades before CRISPR came along. The story starts with mutated fruit flies and ends with rewriting the code of life. The earliest experiments in altering genes weren't elegant at all. In the early 20th century, scientists discovered that they could trigger mutations using radiation and chemicals. In 1927, a geneticist called Hermann Muller blasted fruit flies with X-rays, and their offspring emerged with super strange mutations. They had deformed wings, unusual colors, and disrupted development. It was crude, unpredictable, and often really harmful, but it did prove something incredible: that humans could deliberately change the genetic code. This approach became known as mutation breeding, and despite its intensity, it spread pretty quickly. By the mid-1900s, researchers were using radiation and chemical mutagens on plants to create new crop varieties. Even today, some grains and fruits in our supermarkets trace back to those brute force methods. For example, modern varieties of Ria Red and Ruby Red grapefruit were influenced by radiation to deepen their color. And Golden Promise barley, which was created through gamma irradiation in the 1950s, is still used in brewing beer and whiskey today. The next leap in gene editing came in the 1970s. Scientists were no longer satisfied with random mutations, they wanted more control. In 1972, Paul Berg created the first recombinant DNA molecules, joining pieces of DNA from different organisms. Shortly after that, Herbert Boyer and Stanley Cohen developed techniques to cut DNA with restriction enzymes and insert it into bacteria. So for the first time in history, humans could move genes between organisms. This sparked both awe and fear, because if we could build new organisms, could they escape labs? And could we accidentally create dangerous pathogens? To address these exact questions, scientists convened at the 1975 Asilomar Conference. It was essentially where researchers, lawyers, and journalists came together to decide how far genetic engineering should go. They drafted guidelines that balanced innovation with responsibility. It was a defining moment. Genetic engineering wasn't just science anymore, it was ethics. Hey you! If you love learning about the origin stories of breakthrough technologies, hit the subscribe button now. I drop stories like this every week. By the 1980s and 90s, researchers wanted even more precision. Scientists Mario Capecchi, Oliver Smithies, and Martin Evans developed a method called gene targeting in mice. Using homologous recombination, they could deliberately knock out, replace, or modify individual genes. This led to the creation of knockout mice, genetically engineered animals that transformed medical research, allowing scientists to study cancer, diabetes, immune disorders, and more at the genetic level. Their work earned the 2007 Nobel Prize because it made targeted experiments possible in a way that radiation just never could. But even with these breakthroughs, the process was still slow, expensive, and technically demanding. Gene editing was powerful, but still inaccessible to most. The 2000s introduced new molecular tools: zinc finger nucleases, ZFNs, and later TALENs. TALENs. These engineered proteins could cut DNA at specific locations, offering targeted edits for the first time. Which is to say, for the first time, scientists had designer molecular scissors, if you will. But they came at a high cost. Labs needed years of expertise and a lot of money to use them. Progress was exciting, yes, but it was limited to elite institutions. And then everything changed. In 2012, Jennifer Doudna and Emmanuel Charpentier revealed that a bacterial immune system called CRISPR-Cas9 could be repurposed as a gene editing tool. With just a guide RNA and an enzyme, it could seek out and cut DNA almost anywhere. It was fast, inexpensive, and shockingly easy to use. And basically overnight, gene editing became democratized. Within months, CRISPR was being used on plants, animals, and even human cells. The floodgates had opened and the age of precision biology began. But such power came with consequences. In 2018, Chinese scientist He Jiankui stunned the world by announcing the birth of the first CRISPR-edited babies, twin girls whose genomes he altered to resist HIV. The scientific community responded with absolute outrage. He was condemned, barred from research and imprisoned. The incident made one thing clear: gene editing had stepped out of the lab and into human life long before society was even prepared. Today, CRISPR and its successors are everywhere. Researchers are engineering drought-resistant crops, editing pigs to make their organs compatible with human transplantation, and testing medical treatments for sickle cell disease, muscular dystrophy, and even certain forms of blindness. Yet the technology continues to evolve. New tools like base editors can swap a single DNA letter without breaking the strand, while prime editors, often called CRISPR 2.0, promise even more precise and predictable changes. And now we arrive at the real question. If humanity can rewrite life's code, how far should we go? Should gene editing be limited to curing disease, or could it expand into enhancing intelligence, strength, or even lifespan? I'm really curious to hear your thoughts about this, so let me know in the comments, because this is the ethical crossroads that we stand at today. From X-rayed fruit flies to CRISPR-edited embryos, the history of gene editing is the story of humanity learning to wield the most powerful tool ever created: the ability to shape life itself. And whether this becomes our greatest medical triumph or our greatest cautionary tale is a chapter that is still being written.